I'm Dr. Collin Hobbs. I'm an assistant professor here at Huntington University in biology. And my professional training is as a plant ecologist from Indiana University. It's where I did my PhD. One of my areas that I'm interested in are the genetics of locally rare plant populations. Earlier this spring, since we spring 2015, I was looking at doing summer research, having 2 years behind me here at Huntington. Things are a little bit more relaxed and I have time now to be thinking about actually picking up my research again. And I was talking to Mike Wanis about that, and there was a new opportunity, and this was announced to all of the faculty at Huntington, that there would be a new in-house grant opportunity through the Emeriti Scholars Fund to pay for research that our faculty do. So, along with a number of other faculty, I wrote a proposal for that. Mine was one that was accepted. So the research proposal involved looking at the population genetics of white pine, which is a conifer. It's fairly common in Indiana, but as far as natural populations, there's actually very few isolated natural populations that go back before human settlement or European settlement in Indiana. So we have a situation kind of like this, where you've got an area where the tree, is very common out here. You see a lot of that in that part of its distribution. And here in Indiana, we're like out here, kind of out on the margins of its distribution. And we have some small isolated populations out here. So in my research, what I'm looking at is I'm interested in what's going on with the population genetics out here. Is there a lot of variation? Are there rare genetic traits that are found out there that are not found in the main distribution? And we're also looking at how did these populations arise? Where did they come from? This year's grant was primarily as a pilot study to purchase all the equipment that you need to do this kind of what we call molecular genetics, and there's a lot of equipment involved in that. Also to trial all of the protocols to make sure that they work, and to collect some data, like real data on some samples, to make sure that everything's working, that we can do some basic analyses with that. So basically what we did this year is we went out to a single location in Indiana near Shade State Park over by Crawfordsville, and we sampled needles from 20 different trees. These are native white pines. That population's been there for, who knows, thousands of years. And then we take those needles back to Indiana, so we're starting basically with the needle tissue that we have from these pine trees. And then we're going to do what we call a DNA extraction with this. Basically, you're grinding it up with a lot of harsh chemicals and you're trying to separate the DNA from the needle from all the other compounds, all the phenolics, all of the defense compounds that are found in these needles, so that in the end, mostly what you have is just DNA instead of a little test tube. That's the DNA extraction process. After that, we do something called PCR, which stands for polymerase chain reaction. And in PCR, what you're doing is you're amplifying a region of interest in the organism's genome. From there, we can run some gels. The primary thing we're doing with the gels is just making sure this works. You can check to see if there was amplification of the areas of interest by running a gel. So it's a way of viewing how big those fragments of DNA are. And then after that, we do something called fragment analysis. And what we get back from that is an exact base pair length of the fragments of DNA that we amplified back here. And that tells us something about the genetic diversity in these populations that we started with. So I think another aspect that's great with doing research here at Huntington is that it opens up opportunities for undergraduates to do research. So I had 2 students, 2 biology majors work with me this year doing this research, and they'll be presenting a paper at Indiana Academy of Sciences, a poster, this March. And those are excellent opportunities for them as they're thinking about their future careers in science and where they want to go. I think opportunities like this are really excellent, especially for younger faculty, where you may not have as large of a research record behind you already, to draw on something like that that's coming from your institution. Any of the other faculty who are thinking about doing research, I think would be an excellent opportunity, and they should definitely pursue that.